guys. I just kind of want to go over the cell membrane passive transport notes with you. So let me present them. Okay, so we're talking about how things move across a cell membrane, whether they're moving into the cell or whether they're moving out of the cell. And we're going to focus on passive transport today. So the big question is, how do they move? Do they just magically appear outside, or do they magically push their way through? Do they dig a hole and dig a tunnel? And the last one is one of my favorites here. Do they bribe a protein to let them in? Well, there's two main processes. There's passive diffusion, and then there's passive transport, and then there's active transport. We're going to focus on passive transports. So there's three different types. There's diffusion, facilitated diffusion, and osmosis. Osmosis is just the movement of water. It's a special type of passive transport. Diffusion and facilitated diffusion are where things can move across a membrane without needing extra energy. They just kind of do it on their own. And those are the three ones that we're going to focus on: diffusion, facilitated diffusion, and osmosis. Those are the ones we're going to focus on this week. Then active transport is where you kind of have to use a lot of energy. You got to bribe those proteins in the cell membrane to let things in and out because it needs energy to get them to come in and out. So passive. Again, it's the process that moves things without using energy. So basically, if you got a lot in one area and only a little bit in another, substances are going to move from the high area to the low area until they're equal, until you get an equilibrium. So, for example, I have written on the slide here, 70 molecules outside, only 40 molecules inside the cell. What's in the middle? 55. So the 70 is going to lose 15. The 40 is going to gain 15. The 15 from the 70 is going to move to the inside until they're equal at 55. That's called a concentration gradient, when there's a difference in the amount of something across a space. So the difference between the 70 outside and the 40 inside, that difference across the membrane would be the concentration gradient. And if there exists a concentration gradient, things will move by passive transport, depending on what the molecule is. So if the difference exists, substance will move. They go from high to low until equal. That's it. So the graphic on the screen is showing you the movement of like dye. Say you put yellow food coloring in water, you put a couple drops. By passive transport, the high concentration of that drop, the dye is going to separate the little tiny dye particles, and they're going to move all around until it's all equal. If you have two things, so you got purple on this side and yellow on this side, and you've got a cell membrane in the middle, some of the purples are going to go to that side and some of the yellows are going to go to this side until you get it equal. Again, high to low until equal. So the first one is simple diffusion, just regular diffusion. It's the random movement of molecules with the gradient, with moving from high to low until equal. However, what if there isn't a gradient? What if there isn't a concentration gradient? There is no difference. So are molecules not going to move? No, they're always moving. But if two go out, then two come in so that they make the balance again. If 4 go out, then 4 will come in. So if 4 go out, then 4 will come in. So they're always moving out and in equally. So you have that homeostasis, you have that balance. So there's no gradient. However, things are still moving out. They're just moving out equally. And molecules are either going to push their way through the phospholipids, those like jellyfish little thingies in the phospholipid bilayer, or they're going to go through a channel protein. They're just going to slide through that tunnel. So again, this is what I was talking about. When you put food coloring in a thing of water, they spread out until it's all evenly spread out. And that's diffusion. Notice a little Osmosis Jones there, in case any of you remember that cartoon. Osmosis is the movement of water, and only water, across the membrane from high to low until equal. And we call certain solutions either hypertonic or hypotonic. And it's so they're the hyper and the hypo, they're opposites, and they're measuring like how much water in comparison to a solute there is. So that's a lot of technical talk. But basically, you're looking at the concentration of water. If you have like a real salty solution, the salt is taking up space where water molecules would be. So if you have a cell in salt water, the cell's going to have more water in it than concentration wise than the salt water is, because the salt water is sharing water with salt. So there's less water there in comparison. So if you put a cell inside a hypertonic solution, like on the slide here, that cell is going to give some of its water to the salt water until the concentration of the two waters are equal. So that cell, to give up water, is actually going to shrink a little bit. Here's the example I was just talking about. Okay, so the water's going to move out until it balances the amount of concentration. So say it's like a 4% salt solution. If it's a 4% salt solution, it's only a 96% water solution, where your cell is 100%. So if you have 100 here and 96 here, you're going to lose 2 and gain 2. So it'll be 98, 98. And that your cell is going to shrink a little bit. It's actually called plasmolysis. When your plants wilt, it's because you didn't water them enough, and the soil was so dry comparative to the plant that water actually left the plant to go out into the soil, and that causes your plants to wilt. Hypotonic. So hypotonic is the opposite. It's when you have more water outside the cell. The concentration of water is higher outside the cell. And so the solute, let's say it's, I don't know, like a 100% pure water. And then you have a cell which has 4% glucose, sugar in it. So you have 100% water outside, but you only have 96% water inside the cell because you have a lot of sugar in there. So it ends up, some of the water from outside goes into the cell and makes the cell swell up, get bigger. So when a cell is placed into a hypotonic, the big O makes me think of, oh, it got bigger. And the cell is going to swell up. And sometimes cells will actually swell up so big that they burst if they're animal cells. If they're a plant cell, they're just going to kind of like get bigger and bulkier, but that cell wall doesn't let them burst open. But animal cells, they do. So here's a graphic to show you what happens in a hypotonic solution. These are red blood cells. If you put the red blood cells in distilled water, that there's more actually water concentration in the pure water than it is in the cells. So the cells blow up and they burst. But if you put it in a hypertonic solution, like a saltwater solution, the cells are going to shrivel up because they're going to lose water to the outside to balance that out. Here it is in plants. So you can see the normal cell, how it looks kind of bloated. And then here the membrane actually pulled away from the cell wall and kind of shriveled. All right. What they want is isotonic. Okay. Isotonic is when it's the same concentration outside as inside. So it's a nice balance. Water is still going to move in and out, but there's no overall net movement, meaning more didn't go out than came in, or vice versa. It's equal. And if things are equal and there's no net movement, no overall extra going out or extra coming in, then even though water is moving in and out, we say osmosis is not occurring because osmosis is from high to low. And if there is no high and is no low because they're equal, then there's no osmosis. Just kind of like a tricky question. Facilitated diffusion. That's the last kind of passive transport. So facilitated diffusion uses proteins. They're called carrier proteins or transport proteins, and they help carry bigger things across the membrane that can't fit through the channel proteins and can't fit between the phospholipids. So they help carry the big things. They still go with the gradient from high to low because everything in here in passive transport is high to low. So sugar, amino acids, these things are too big to fit through those channel proteins, so they have to go with a carrier or a transport protein. So you can see these molecules can go through, but this molecule here, even though it's the same size, they should have used a different shape and a bigger one for this picture, but whatever. It goes through the carrier protein, and the carrier protein kind of like squeezes it and pushes it to the other side. So these two ways, if it went through the phospholipids or the carrier or the channel, it would be simple diffusion. But if it went through the carrier protein, then we're going to call it facilitated diffusion. And what's kind of cool is the carrier transport proteins have specially designed openings so that the specific thing that it carries through is the only thing that fits there. So you can see like the sugar would fit into this one, an amino acid would fit into this one, but the amino acid wouldn't fit into the sugar carrier protein. It only fits into the amino acid carrier protein. So it's kind of cool. All right. So that's our stuff for today. The passive transport, again, high, the things you need to definitely remember, high to low until equal, and diffusion, facilitated diffusion, and osmosis, the three different types. So if you need to help rewind that and watch again, go for it. Otherwise, that's it. So, all right, let me try and figure out how to stop this.